Welcome to this lecture on understanding FDA. The modern-day FDA was founded in 1906 with the passing of the Pure Food and Drug Act, which enforced accurate and truthful labeling of products. Product safety was still not regulated until 1938, after the sulfanilamide crisis of 1937 where 107 people, mostly children, died from a toxic preparation. The Federal Food, Drug and Cosmetic Act came into being, requiring safety to be proven before marketing. Product efficacy was addressed in the 1962 amendments to FDCA for drugs. Device regulation was implemented by the Medical Device Amendments Act of 1976. The FDA Modernization Act of 1997 brought reforms including accelerated reviews and regulation of unapproved drug uses. This evolution reflects society's increasing expectations for product safety and efficacy. FDA's mission encompasses several interconnected goals. Protect the public health by assuring the safety, efficacy, and security of human and veterinary drugs, biologics, medical devices, food supply, cosmetics, and radiation-emitting products. Advance public health by helping speed innovations that make medicines more effective, safer, and more affordable, and by helping the public get accurate, science-based information to maintain and improve health. The agency serves as gatekeeper to the market, ensuring that claims made about products accurately reflect benefits and risks. FDA provides specific wording regarding claims, indications, and safety through approved labeling. Understanding this mission helps companies anticipate FDA expectations and priorities. FDA organization includes the Office of the Commissioner overseeing policy planning, administration, and finance, plus specialized offices for combination products and orphan products. The Center for Drug Evaluation and Research regulates all human drugs and certain biological products not regulated by CBR. The Center for Biologics Evaluation and Research regulates products derived from living sources including cellular products, vaccines, blood products, and gene therapies. The Center for Devices and Radiological Health regulates all medical devices including radiation-emitting products and the Office of In Vitro Diagnostics. Additional centers cover food safety, veterinary medicine, and toxicological research. Understanding this structure helps companies navigate regulatory interactions. FDA regulates an extensive range of products affecting public health. Blood-related biologics and blood substitutes fall under FDA jurisdiction. Drugs including biological large molecule drugs require FDA review and approval. Medical devices including diagnostics must meet FDA requirements appropriate to their classification. Foods except meat and poultry are regulated by FDA with USDA handling the exceptions. Cosmetics and radiation-emitting electronic products also fall under FDA authority. Veterinary products complete the scope of FDA oversight. Understanding what FDA regulates, and what it does not regulate, helps companies identify the appropriate regulatory pathway and agency interactions required for their products. Understanding what FDA does not regulate is equally important. Consumer advertising falls under Federal Trade Commission jurisdiction, while FDA regulates prescription drug promotion to healthcare professionals. Alcohol is regulated by the Bureau of Alcohol, Tobacco, and Firearms under Treasury. Consumer products generally fall under the Consumer Product Safety Commission. Illegal drugs are regulated by the DEA under the Justice Department. Health insurance is overseen by CMS. Meat and poultry are regulated by USDA, while FDA handles other foods. Pesticides and water quality fall under EPA jurisdiction. This division of regulatory authority means companies may need to interact with multiple agencies depending on their products. The FDA-industry relationship has historically been characterized by tension due to very different priorities, FDA focuses on safety while industry emphasizes speed to market. The Prescription Drug User Fee Act has reduced drug review times in exchange for substantial fees paid by industry. The Medical Device User Fee and Modernization Act similarly reduced device review times. Recent FDA efforts to work more collaboratively with industry include assistance with applications, user resources through the FDA website, simplifying and modernizing the critical path to approval, and efforts to fast-track certain applications. These initiatives reflect recognition that efficient review processes serve both public health and innovation goals. Because of increasing global trade in food and medical products, and differences in country regulations, ongoing efforts aim to harmonize requirements for review and approval across jurisdictions. FDA is actively involved in the International Council for Harmonization to make international regulatory processes for biomedical products more uniform and efficient. ICH brings together regulatory authorities from major markets including US, EU, and Japan, along with industry representatives. Guidelines created and published by ICH are adopted by individual national regulatory bodies. Harmonization reduces duplicative requirements and facilitates global development programs while maintaining appropriate standards for safety and efficacy. An investigational new drug application must be filed to conduct clinical studies on an unapproved drug or on an already approved drug for unapproved indications or new doses and forms. Two types of INDs exist: commercial INDs providing the full version for products intended for eventual marketing, and research INDs providing abbreviated versions for investigator-initiated studies. In general, clinical work can be initiated 30 days after FDA receipt of the IND unless FDA places a clinical hold. Notably, no specific approval letter is sent, the sponsor proceeds unless notified otherwise. However, best practice is to notify FDA of clinical trial initiation. The IND remains active throughout clinical development with required safety updates. The IND includes several critical sections. The General Investigation Plan provides a summary overview of the new compound and plan development. The investigator's brochure contains all clinical, non-clinical, and CMC information to support further development, including scientific rationale. The clinical protocol details how clinical trials will be carried out, including proposed endpoints and biostatistics analyses. The CMC section describes the product substance, analytical methods, and manufacturing process overview. Pharmacology and toxicity information includes single and repeat dose toxicity studies typically required by FDA. Each section serves specific purposes in demonstrating readiness for human testing. Several key FDA meetings guide drug development. The pre-IND meeting discusses design and scope of proposed clinical trials before beginning human testing. It's a formal meeting with minutes, focused on data and scientific methodology, with agreement sought in writing. The end of Phase 2 meeting presents proof of efficacy, safety, and other data to support Phase 3 initiation, with Phase 3 protocols and endpoints agreed upon. Pre-NDA meetings discuss submission content, format, and any remaining questions before filing. Each meeting follows established procedures for requesting, preparing, and documenting outcomes. Written agreement provides important protection if questions arise later about what was agreed. The new drug application contains comprehensive documentation. The application summary includes proposed package insert, indication summary, pharmacology summary, clinical summary, and CMC summary. The CMC section describes product characteristics, analytical methods, and manufacturing process in detail. Pharmacology and toxicology provides description of all animal and in vitro studies conducted. Human pharmacokinetics and bioavailability covers all Phase 1 pharmacokinetic and pharmacodynamic studies. Clinical data includes all clinical investigations demonstrating efficacy and safety, integrated summaries of effectiveness and safety, and statistical analyses. Safety updates provide new safety information collected while the application is under review. Biosimilars are highly similar to already approved biological reference products but not considered biologic generics. There must be no clinically meaningful differences from the reference product, same mechanism of action, dosage form, strength, route of administration, and indications. Importantly, no substitution of reference product is allowed without physician involvement for biosimilars. Interchangeables meet the biosimilar standard plus produce the same clinical result as the reference product in any given patient. They are considered exactly the same as the innovator product, and reference product may be substituted without physician permission. The Biologics Price Competition and Innovation Act established these pathways under PACA in 2010. FDA offers several accelerated pathways for products addressing serious conditions. Fast Track is a designation for treatment of serious or life-threatening conditions with no current treatment, requested early in development, allowing greater FDA interaction and rolling review. Breakthrough therapy requires demonstration of substantial improvement over existing therapies, with senior FDA commitment and intensive guidance. Accelerated approval is an approval pathway allowing earlier approval based on surrogate endpoints predicting clinical benefit, typically with required confirmatory trials. Priority review provides 6-month review versus 10-month standard for products offering major treatment advances. Each pathway serves different circumstances and may be combined. FDA classifies medical devices based on risk level, which determines regulatory requirements. Class 1 devices are subject to least regulatory control because they present minimal potential harm to users. Most are exempt from pre-market notification, with examples including elastic bandages and handheld surgical instruments. Class 2 devices usually require 510(k) pre-market notification and are subject to special controls such as performance standards. Examples include infusion pumps and powered wheelchairs. Class 3 devices are most strictly regulated because they support or sustain human life, usually requiring PMA. Examples include coronary stents, heart valves, and hyaluronic acid injectables. Classification determines the regulatory pathway and evidence requirements. The 510(k) is a pre-market submission demonstrating the device is safe, effective, and not subject to pre-market approval. It demonstrates substantial equivalence to a device already on the market, predicate device, showing same intended use and technological characteristics. Generally little or no clinical data is required, though FDA increasingly requests clinical evidence for certain device types. Several 510(k) types exist, traditional for complete submissions, special for modifications of own prior devices, abbreviated for devices conforming to special controls, and de novo for novel low-risk devices without predicates. Importantly, 510(k) products receive clearance rather than approval, an important legal distinction. The pre-market approval application is the device equivalent of a drug NDA or biologic BLA in complexity and data requirements. It represents a private license to the applicant, allowing the device to be marketed, and provides reasonable assurance through detailed science and clinical trial results that the new device is safe and effective. Clinical trials are required to generate the evidence supporting PMA. FDA requires a minimum of 180 days to review before approval or non-approval decision, though actual review often takes longer. PMA types include traditional for one-time complete submission, modular for rolling submissions of completed sections, streamlined for well-known technologies, and product development protocol for agreed development criteria. Pharmacogenomics, also called pharmacogenetics, studies how genes affect a person's response to drugs. It combines pharmacology and genomics to develop medications and dosing tailored to genetic makeup. Pharmacogenetic tests help identify target populations for selection of certain therapeutics and compare interindividual differences in genomic patterns. These tests could identify populations most likely to respond to a drug or most likely to have toxic reactions. Co-development pathways for diagnostics and drugs have been established by FDA to accommodate two different but synergistic products. Companion diagnostics provide the right drug to the right patient at the right time, representing a key element of personalized medicine. The Office of Combination Products was created in 2002 to help guide innovative combination biomedical products that cross over defined FDA review divisions. Primary regulatory oversight remains with the three product centers, CDR, CBR, and CDRH, with OCP assigning a lead center that will have primary jurisdiction for review based on primary mode of action among other criteria. OCP develops policy for combination products, ensures timely and effective pre-market review, and ensures consistency and appropriateness across the agency. As combination products become more common, OCP's role in coordinating review and providing guidance to sponsors has become increasingly important. This week examined FDA organization, mission, and regulatory pathways. FDA's mission balances protecting public health through safety assurance with advancing health through innovation support. Understanding FDA's organizational structure helps companies navigate regulatory interactions appropriately. Multiple pathways exist for drugs through IND to NDA and for devices through 510(k), or PMA, with classification determining requirements. Accelerated pathways including Fast Track, Breakthrough Therapy, Accelerated Approval, and Priority Review serve products addressing serious conditions. Biosimilar and interchangeable pathways enable competition in biologics markets. Combination products and pharmacogenomics represent growing areas requiring specialized regulatory approaches. Next week we examine technology transfer to manufacturing operations.